So our next speaker will be Benjamin Dauphin from WSL, and he will talk about genomic signatures of drought adaptation in Fagaceae.
Good morning. Thanks to the organizers. Let's start, folks. So climate change is global, but is discontinuous through times and space. And based on these temperature anomalies in the last 140 years, we had cooler and warmer conditions at different time periods, but clearly in the last 2 decades we had an acceleration of in temperature increases. And what is, I think, very interesting here is to look at different long-term effects versus extreme events like heat waves. And this is very important when we try to capture climatic pressures. Another main concern is about the drought events, and here are the past drought events in Europe based on Cook et al. Studies. And as you can see here, the brownish colors mean the severity of the drought. And again, is quite discontinuous actually through space. So in 1921, the south countries were not really affected by the droughts, but the continental Europe was. And for other more ancient events, it was the reverse pattern. So more recently, we had this extreme drought event in 2018. During summer and fall in Europe, and we had these cases of dieback on beech trees. Here, the first, it's a Swiss forest in Jura Mountains where we had really trees suffering of drought and even dying. And a year later, in Marburg Open Forest University, we had this lag also. That we can observe on dieback tree species. So one way to cope with the rapid environmental change is to adapt to changing conditions. And it has been shown that the standing genetic variation is a predominant source of these beneficial mutations because they can be fixed quicker compared to new mutation arising spontaneously into the populations. And also it has been hypothesized that the long-lived forest trees may have difficulty to follow the fast pace of climate change because they have a long generation time and they can suffer of this adaptational lag, so they need time to change their allele composition. Something important to keep in mind is we have the persistence of potentially maladapted old trees, so they can still spread out by pollen and seeds, suited genotypes for past conditions. So our research questions so far are, do beech and oak, so it's really Fagaceae-based analysis, experience contrasting selection pressures in space and time? Are they showing signal of selection through climate and soil-related droughts, because the drought can be characterized as different environmental components? And finally, we try to maybe generalize something about the genetic mechanisms underlying climate adaptation of trees in Europe. So we use genotype environment association analysis to search for significant correlations between environment and genetics. And here, each dot represents a population, and we are really looking at these significant linear regressions between, let's say, temperature and allele frequency, or the severity of drought versus allele frequency. So we used 3 main environmental data sets to characterize the environment. The first is the soil greedy data set using 2 different variables. This is the available water capacities at 2 different soil horizons, with superficial horizons versus the deep horizons. And the goal is to track signals of edaphic-induced drought. The second package is the topography, using the Copernicus European Digital Elevation Model. And here we want rather to capture the drainage-induced drought, so the drainage effect, plus the radiation and the macro topography in terms of protection and wind desiccation. And the last one is a climate data set using CHELSA data. And we have like 3 subdivisions with climatology, with the mean temperature, minimum temperature, maximum temperature, average temperature, and precipitation for the 4 seasons. The bioclimes and the drought, 4 variables plus 2 frost variables. So I don't want to go too much in detail, but we used the GenTree sampling, of course, by pairs. And so here you have for beach, the sampling scheme. So each dot represents a pair, actually. And for oak, we have more or less the same number of populations. And at the bottom, you can see what is the what is the climatic envelope that is captured by GenTree sites. So we have a pretty good representation of the species range, actually, which is very good for this selection analysis. So just to get a first clue about the ecological group based on this environmental data, again, you have on the left beach and on the right oak. Clearly, we don't have perfectly matched with geography, meaning that the German populations for Fagus pop up in different ecological groups. We have 7 in beech, 6 in oak. For the French populations, we have 2 different ecological groups. And for beech, it's a little bit better. Only the Italian populations that are split. But then there is no really similarity in terms of geographical distances. So like we have Norway that is similar to Switzerland and so on and so forth. So this picture is very, very important when we look at environmental selection, because we don't have a covariance of historical population demography. So that's very good to have that. As Katerina said before, it's very challenging to use the environmental contrast because if we have, let's say, 100 meters of elevational contrast in Switzerland, in Spain, and in UK, we don't have the same selection pressures, so it's very difficult to compare the contrast. Rather, we can have a look at the gradient across populations, or at the final scale, look at the declines. So here you have the populations that are ordered by latitude, and you can see for each pair, in Switzerland, for example, we have the first line and the second. Here's the potential direct solar radiation, so just one topographic variable. So in Italy, for example, we have a quite important line, but in Lithuania or in France, not that much. So we can compare at different scale the selection pressures using this kind of data. I don't want to go too much in detail, but we used the exome-capture data set that were generated in GenTree, and but this time we used a reference genome to look at the SNPs that we could get, and we performed a principal component analysis to look at what is the overall genetic structure, and this information is used later for genotype environment association analysis. So remember, we try to look at these significant linear regressions, accounting for population structure, which is very important, and we ended up with this number of significant associations. And what is important, what is interesting in my opinion, it's to compare this number of associations with the average beta, so the slope of this regression, meaning the strength of the response we get. For the soil data, we have only 21 significant associations, but we have a quite high beta compared to other like topography like variables where the beta is almost nil. Also, for climatology, for example, we have many significant associations with quite high beta, and in this case, it behaves like a bit like the soil results, but in other cases, the beta is quite low, and maybe probably we have a smaller effect size in that responses. So another way to look at these first preliminary results is to look at the behavior of each environmental variable using a combination matrix. So for example, you have 3 different bioclimes, and they share this SNP, and you can represent it like each bioclim has a dot, a black dot that are connected with edges. But if the SNP is shared only by bioclim 2 and bioclim 3, then it's getting like that. You have the black dots that are connected, but the first bioclim 1, which has no significant association, stays a gray dot, okay, and so on and so forth. So it looks like that for the bioclims. You have all the variables on the left, and at the top you have the number of shared SNPs across all variables, and at the right panel you have the number of significant associations. So it just give us a bit of what is the redundancy in terms of signal plus the number of associations we have. And if you don't get the graph, just you have a shortcut at the bottom. It says that just we have a stronger signal for beach, in this case in precipitation-related bioclims. So most of the significant associations are precipitation-related bioclims. For HAWK, the strongest signal is in temperature-related variables, so the reverse pattern, and it's quite striking actually. We have no signal for precipitation-based data. Now if we focus on drought and frost for beach, we have again a signal for precipitation, but also for more sophisticated humidity indices like hydrothermic coefficient or relative humidity. And again, it's quite consistent with the pattern we capture with climatology data. For HAWK, this time we have a signal in drought-related variables, so with the relative humidity and hydrothermic coefficients. But again, we have nothing captured from the precipitation data, meaning that we need more sophisticated variables to really capture the signal. So it's quite interesting to look at these different contrasting patterns. But this is just at the SNP level, and of course we want to know more about the genes, and we use the HAWK reference genome to test if we have enrichment for associated SNPs in genic regions. So you have again the group of environmental factors: soil, topography, climatology, bioclims, drought, and frost. And within we have several variables. Keep it in mind. So the blue line here represents the number of SNPs that are significantly associated with one of the two soil variables. And the red lines are the SNPs that are in the intergenic regions. And at the background, you have an empirical distribution of randomly resampled SNPs in the full data set. And from this one, we can calculate an empirical p-value to know if we have a significant deviation from what is expected randomly. So for soil and frost, we have no significant deviation. But for topography, climatology, bioclim, and drought, it seems that there is some forces going on, and we have more SNPs falling in the genic space, so, which is quite interesting. And now we want to know what are those genes that are under this environmental selection. So just to sum up this, again, very preliminary results. We have contrasting signal pressures in beech and oak for temperature and precipitation variable. And it's quite interesting because it's still this hypothesis whether oak or beech is more adapted or will cope with climate change in a different way. And then we have a significant enrichment for SNPs in genic regions associated with topographic, climate, and drought variables. That's a statement. We'll see then if those are relevant genes involved in these variables. And finally, it's a bit provocative, but well, my take-home message is because we have this environmental gradient across south to north populations, there is no straightforward assisted gene flow from south to north, because sometimes, as I've shown, declines really shows that you can be in south but still have quite cool conditions compared to continental Alps or Central Europe. So I think we have to put a bit more effort, so mostly for population geneticists, into characterization of environments. And the outlook, again, it's a big team behind me. So we want to conduct this analysis at the gene level. We want to test the convergence and divergence of genes network in response to drought or frost conditions, because it's one of the main topic in Fagaceae. And we want to link genotype-phenotype association analysis to investigate gene function underlying dendrophenotypic traits for drought or even frost resistance. So we don't really know if we could link the two data sets, but really the goal is to track at the phenotypic level if what we are doing makes sense. Of course, I'm just a piece of a long chain. I want to thank all GenTree partners that were part of the project. And yeah, our big loss, I realized this morning that yeah, we have Petit Fidi here. And I thank you very much for your attention.
Time for questions?
Tanja Pyhäjärvi from University of Oulu. I wanted to ask about this enrichment analysis for the genic SNPs, and I recycled the question that Andrew posed to me. Did you control for the heterozygosity? Because the heterozygosity could be different in the genic and non-genic regions.
No.
Okay, I think you should probably do it.
Okay.
François Lefebvre from INRAE. Thank you for your presentation. You had a lot of results, first presented with the number of associated significant associations and the beta value, and you had different things, and then many results in terms of number of significant associations. And actually, I don't clearly, and there are big differences, but I don't clearly see what does it tell us, actually, the number of significant associations. Does it tell us something about the genetic architecture of the trait under selection, or does it tell us something about the selection process itself? But yeah, how do you interpret this number of significant association? What does it mean for you?
So for me, it's of course the first step of the whole analysis, and what we know from other data set is even if we have, for example, correlated variables for climatology, we can still have different SNPs that are significantly associated even with correlated environmental variables. So that's why we have to make this exhaustive analysis, and then we have to conduct exactly the same at the gene level. So meaning that we really categorize which SNPs belongs to which gene, and then we look at which one are shared among variables, or also soil-induced drought versus climatic-induced drought. And that's the whole trick, actually. It's to make the comparison between variables at the gene level and then to make the comparison between species. But we have to know if we have 5 significant associated SNPs or 500. It's a very, very first step we have to make.
There is a difference, but then what means this difference?
So for the bioclimes, we can just group what is temperature-driven versus precipitation-driven, and then we can refine the picture. For example, for the drought event, if we have to use a drought event, that is a drought variable, that is calculated from precipitation or from temperature, or both together. So, first clues.
But this is all based on GenTree samples, right? You did a similar study as well, Christian, some years ago, only in Switzerland. When you compare the results that you obtained, for example, in petraea in Switzerland, and then on the European scale in GenTree, in terms of, for example, outliers, do you see any similarities? Or?
From the genes I cannot say yet, but of course from the environmental factors that have the highest signal, we had much more precipitation signals in Switzerland, yes, yeah, for petraea.
Because the gradient is very different, so yeah. And the precipitation at local scale is very difficult to capture from models.
It was just a complementary question to the one that Tanya did and Andrew did before Tanya. It's like, how you check out also for differences in LD between genic and non-genic regions, because there could be also, if they have different levels of linkage disequilibrium, it will also show the different number of associations.
Yeah.
So, how you check for these differences? So?
They are not checked yet.
Okay.
It's a pure statistical test where you randomly resample the full SNP set, and then you see if you have a significant deviation based on this empirical p-value. But then, of course, that's just a starter, and we want to look at what is the... And also within the genic regions is very interesting because it's a high reference genome, a high quality reference genome. We can really look at which one are CDS and which are really the type of categorization we can test. That's on the roadmap. But